So as we begin our section on erythrocytes, we'll see that they have two principal functions. First, they carry oxygen from the lungs to the cell tissues, and then they'll pick up carbon dioxide from these tissues and bring it back to the lungs. Now, an insufficient supply of red blood cells can kill someone in a few minutes due to lack of oxygen in the tissues. Not that insufficient supply of all the other formed elements won't cause problems, but that'll take a few days. This micrograph here shows us the red blood cell's shape. You'll notice that there are no organelles visible here because there are no organelles like mitochondria or ribosomes and such inside the red blood cells. So the cell itself is disc-shaped with a thick rim. It's about 7.5 micrometers in diameter and about 2 micrometers thick at the rim. It loses nearly all its organelles during development. It lacks mitochondria, which means that it is solely able to do anaerobic fermentation to produce its ATP. Now this prevents it from consuming the oxygen that it carries in aerobic metabolism. It lacks a nucleus and DNA, and so it doesn't perform any protein synthesis. So the lifespan of a red blood cell is going to be fairly short. The blood type of an individual is determined by the surface markers. There are glycoproteins and glycolipids. There are cytoskeletal proteins inside called spectrin and actin, and they give the membrane durability and resistance and hold the red blood cell's shape. It allows the red blood cells to stretch and bend as they squeeze through the small capillaries, because the very smallest capillaries are smaller in diameter than the red blood cells. So they'll squeeze and bend, and then they'll reform their shape, just like an inner tube, say, that was filled with air. So the primary function of the red blood cell is obviously gas transport. They can transport a lot of gas because this increased surface area to volume ratio allows diffusion of carbon dioxide and oxygen into and out of the cell. Especially, it allows increased contact of oxygen with the hemoglobin molecules that we'll see inside the red blood cells. 33% of the cytoplasm inside the red blood cells is hemoglobin. That amounts to 280 million hemoglobin molecules in one red blood cell. Hemoglobin is involved in both oxygen delivery to the tissues and carbon dioxide transport back to the lungs. We'll see how that works out in some upcoming chapters. We'll also find inside the red blood cell an enzyme called carbonic anhydrase. It produces carbonic acid from CO2 and water. As we'll see later, it has an important role in gas transport as well as pH balance. Hemoglobin consists of 4 protein chains, or globins, and 4 heme groups. We have 2 alpha chains and 2 beta chains, and then these heme groups. The heme groups are a non-protein moiety that bind the oxygen. There's a ferrous ion, Fe2+, and oxygen, being very electronegative, is drawn into this center. When CO2 is transported away from the tissues and to the lungs, about 5% of it is bound to the globin moieties of hemoglobin. So oxygen binds to the heme groups. However, CO2, when it's transported away, binds to other moieties of the globins. Now, there are several different types of hemoglobin. There's 2 types of adult hemoglobin, and then there's fetal hemoglobin. Now, fetal hemoglobin has 2 gamma chains instead of the beta chains, and they're the same shape and length and so on and so forth, but with a different amino acid sequence. So they bind oxygen much more tightly than the adult hemoglobin version. And this is important because the fetus needs to be able to extract oxygen from the maternal blood. So the red blood cell count and hemoglobin concentration are big indicators of the amount of oxygen that blood can carry. We've already looked at hematocrit, or packed cell volume, or PCV, which is the percentage of the whole blood which is composed of red blood cells. In men, that's usually 42 to 52% cells. In women, it's usually 37 to 48%. The hemoglobin concentration is also different between men and women. 13 to 18 grams per deciliter in men and 12 to 16 in women. The red blood cell count itself is 4.6 to 6.2 million per microliter. And in women, we'll see 4.2 to 5.4 million per microliter. Now, values are lower in women for a number of different reasons. First of all, red blood cell production is stimulated by androgens, and men have more androgens, and thus that would stimulate more production of red blood cells. Second of all, women of reproductive age have periodic menstrual losses. And third, hematocrit is inversely proportional to the percentage of body fat, and women in general have higher levels of body fat. Now, there may be some evolutionary reasoning for this, in the fact that males probably fight more often than females, and thus they suffer more injuries, and so it's of benefit to have a higher rate of red blood cell production. During fighting, androgen levels would be higher, which would stimulate more red blood cell production. Now, 2.5 million red blood cells are produced every second. The average lifespan of a red blood cell is about 120 days, and their development takes 3 to 5 days. What happens is they have a reduction in cell size, and then an increase in cell number through mitosis, and then they'll synthesize the hemoglobin and lose the nucleus. That series of developmental steps can be viewed here in the figure above. The first committed cell is the erythrocyte colony forming unit. That's here, the erythrocyte CFU. It has receptors for erythropoietin. Erythropoietin is produced by the kidneys. We'll shorthand erythropoietin as EPO. Erythroblasts, or normal blasts, are going to multiply and produce more and more red blood cells and synthesize hemoglobin. The nucleus is then discarded to form a reticulocyte that's named for the fine network of endoplasmic reticulum inside. 0.5 to 1.5% of the circulating red blood cells are reticulocytes. After they've become reticulocytes, they can develop into erythrocytes. So if there's an accelerated rate of blood loss due to injury, then there's going to be an accelerated rate of erythropoiesis. This leads to an increasing number of reticulocytes as the bone marrow is in such a hurry to release them into circulation to replace the loss of mature cells. So iron is the only key nutritional requirement for erythropoiesis. It's lost daily through the urine, feces, and bleeding. In women, we'll see about 1.7 milligrams per day, and in men, only about 0.9. Women have higher losses because of menstrual cycling. There's a low absorption rate of iron, and it requires consumption of about 5 to 20 milligrams per day. Dietary iron is ferric, or FeF+, and ferrous, Fe2+. Stomach acid is going to convert ferric iron into ferrous iron, which is absorbable. So that means we ingest a mixture of both ferric and ferrous iron. The stomach acid will convert any of the ferric iron into the absorbable form of ferrous iron. Now, there's a complex series of events for iron metabolism that you can see in this figure. There's no need to memorize all the different steps here, but just be aware that it's converted into an absorbable form in the stomach and then absorbed into the blood plasma, and then some of the iron is stored in the liver, and the remaining is distributed to other organs. Another couple of key nutrients needed for erythropoiesis are vitamin B12 and folic acid, and vitamin C with copper. B12 is going to allow rapid cell division and DNA synthesis that's necessary for rapid erythropoiesis. Vitamin C and copper are cofactors for the enzymes that are necessary for the synthesis of hemoglobin. Copper is going to be transported in the blood by an alpha globulin called ceruloplasmin. When we see a drop in red blood cell count, it'll cause hypoxemia, or low oxygen in the blood in the kidney. The kidney will then produce erythropoietin, which stimulates the bone marrow, and the red blood cell count will increase within 3 to 4 days. The figure on the right demonstrates this series of events. Inadequate oxygen transport is sensed by the liver and kidneys, which secrete erythropoietin, stimulating the red bone marrow and accelerating erythropoiesis, thus increasing red blood cell count. The stimuli for increasing erythropoiesis is predominantly hypoxemia, which is low oxygen levels in the blood. This can happen at high altitude with increase in exercise level and loss of lung tissue due to emphysema. When red blood cells lyse in the narrow channels of the spleen, macrophages are going to digest the membrane bits and separate the heme from the globin. The globins are then hydrolyzed into amino acids, and the iron is removed from them. The heme pigment is then converted to biliverdin, which is a green pigment. Biliverdin is then converted to bilirubin, which is a yellow pigment, and then it's released into the blood plasma. This is why we see yellow urine, because of the bilirubin. The liver is going to remove more bilirubin and secrete it into the bile. Bile is concentrated in the gallbladder, which is going to be released into the small intestine. Bacteria will create urobilinogen from this, which is what contributes to the brown color that we see in feces. The figure to the right illustrates erythrocyte death and disposal. Here we see expired erythrocytes are being broken up in the liver and spleen after about a 120-day lifespan. The cell fragments are going to be phagocytosed, and then the hemoglobin degraded. This is the series of hemoglobin degradation. Perhaps now is a good time for a diagram. Close your books and close your notes and see what you can do to produce a diagram that illustrates the sequence of events for erythropoiesis. Then spend a moment looking at how erythrocyte homeostasis is acquired. And also, what are the steps in death and disposal of red blood cells? Do what you can without looking at your notes. See what you can remember, and then look in your notes, close your notes, see what you can remember again. This will create curiosity in your brain and thus make it more adept at holding on to information. Now, before we complete the section on erythrocytes, let's look at some erythrocyte disorders. There's two primary disorders, having an excess of red blood cells or not enough red blood cells. Polycythemia is the case of having excess red blood cells. There are two types of polycythemia. There's primary polycythemia, which is a result of cancer in the erythropoietic cell line in the red bone marrow. The red blood cell count can get as high as 11 million red blood cells per microliter. That's a hematocrit of 80%. Secondary polycythemia comes from any other source, like dehydration, emphysema, high altitude, or physical conditioning. Anything that creates a sense of hypoxemia. And that can drag the red blood cell up to 8 million per microliter. The dangers of polycythemia is that we'll see an increased blood volume because you have increased dissolved solutes pulling more fluid into the blood. That will increase blood pressure, or we could just see increased blood viscosity. This can lead to embolism, stroke, and heart failure. The opposite case is anemia. Anemia is not having enough oxygen delivered in the blood. The cause of anemia can fall into three categories: either inadequate erythropoiesis or hemoglobin synthesis. This can result from kidney failure, which is going to produce insufficient erythropoietin. An iron deficiency can cause anemia. An inadequate vitamin B12, which is going to inhibit the rate of erythrocyte production. And then hypoplastic anemia, which is where we just see erythropoiesis slowing for some reason, or aplastic, which means complete cessation of erythropoiesis. There's also hemorrhagic anemias that result from bleeding, loss of red blood cells and thus oxygen carrying capacity because of a big bleed, or hemolytic anemias, which are from red blood cell destruction. The consequences of anemia are that there's hypoxic tissue and possibly necrosis or death of that tissue. On the lighter end, we see that the patient is very lethargic. There's a shortness of breath upon even a little bit of exertion. Now, if there's necrosis in the brain, heart, or kidney, this can be life-threatening. The blood osmolarity can be reduced because there's not enough formed elements, and thus the blood viscosity could be low. The heart races and the blood pressure drops, and cardiac failure may result from this. One well-known anemia disease is sickle-cell disease. This is a hereditary hemoglobin defect that can occur mostly among people of African descent. It's caused by a recessive allele that modifies the structure of the hemoglobin. It'll become hemoglobin S. It differs only in one amino acid in the beta chain. And because of this one amino acid, it doesn't bind oxygen as well as the regular hemoglobin. Red blood cells are going to become rigid and sticky and pointed at the ends, and that causes them to clump together and block small blood vessels, which causes intense pain as well as a multitude of other effects. It can lead to kidney failure, heart failure, stroke, rheumatism, or paralysis. Sickle cell disease is considered protective in the event of malaria because the formation of sickle cells prevents the life cycle of the malaria parasite. That's why the sickle cell gene has been maintained in African populations. That brings us to the end of our section on erythrocytes. Spend some time with those diagrams. Sort out the material of this chapter before you move on to the next section, where we'll discuss the various blood types.